Think about the basic math of human reproduction. A child inherits exactly half of their genetic material from their mother and half from their father. If human cells simply combined their DNA during fertilization, the resulting child would have twice as many chromosomes as their parents. Within a few generations, the genetic code would become so overloaded it would break down. This diagram shows the biological solution. It is a specialized process called reductional division. To prevent the genome from doubling, a starting diploid cell, marked 2N with 46 chromosomes, must divide that number in half, creating haploid cells, N, with 23 chromosomes each. This exact terminology, diploid to haploid and reductional division, is how your NCERT and WBCHSE Class 11 textbooks officially define the start of meiosis. You will need these specific terms to build your foundation. Before tracking how chromosomes divide, we must clear up a common exam trap: the difference between a chromosome and a chromatid. During DNA replication, the physical mass of the DNA doubles, creating this familiar X shape. Even though it looks like two pieces, as long as those two sister chromatids are held together by a single centromere, it is still counted as exactly one chromosome. Meiosis requires the cell to manage both the physical mass of these duplicated chromatids and the total chromosome count. It accomplishes this through a highly calculated two-step reduction to maintain the genetic stability of our species. That first step is meiosis 1, and it begins with prophase 1. Because the cell has to pair up and shuffle the genetic material, this is the longest and most complex phase. You can track its 5 distinct substages using a simple mnemonic. Little zebras play during daylight. Little stands for leptotene, where the chromosomes condense and become visible. Zebras is for zygotene. Here, homologous chromosomes, the matching pair you got from each parent, find each other, zipping together in a process called synapsis to form a bivalent or tetrad. Play is pachytene, the critical moment of crossing over. Recombination nodules form, allowing inner non-sister chromatids to physically cut and swap DNA segments. During is diplotene. The zipped chromosomes pull away, revealing distinct X-shaped tethers called chiasmata, where DNA was successfully exchanged. Finally, daylight stands for dikinesis. The nuclear envelope completely dissolves, the spindle fibers fully organize, and the cell is ready to actually separate these newly shuffled chromosomes. If you get a descriptive long answer question on your exam, this sequence of 5 stages is exactly where the examiners will look to assign marks. Because of the precise physical swapping during pachytene, the resulting chromosomes are entirely unique. This mechanism is the reason why two siblings, coming from the exact same parents, look completely different. In metaphase 1, homologous pairs straddle the equator of the cell. They line up as partners, randomly facing either pole, adding genetic variety. During anaphase 1, spindle fibers pull back, separating whole chromosomes to opposite poles. Sister chromatids stay attached at their centromeres. We now have two cells with half the normal chromosome count. They are haploid. But remember that X shape? Each chromosome still carries double the DNA mass. We need a second round of division, meiosis 2, which functions almost exactly like regular mitosis, to separate those sister chromatids. By passing any DNA replication, these cells move straight to anaphase 2. Here, the spindle fibers finally snap the centromeres. Individual sister chromatids are pulled to the opposite ends of the cell. The final tally gives us 4 haploid cells. Because of the crossing over and independent assortment we tracked earlier, None of these 4 cells are identical to each other, and none are identical to the original parent cell. This 2-step sequence is known biologically as the reduction equation. Meiosis I reduces the chromosome count, and meiosis II equates the DNA mass, creating perfectly balanced gametes ready for fertilization. Zooming out from the cellular mechanics, meiosis serves 3 distinct roles in biology. It restores the correct chromosome number across generations, it guarantees genetic variation within offspring, and through that variation, it provides the required raw material for natural selection and evolution. When you sit for your Class 11 board exams, these 3 specific outcomes, maintaining chromosome number, variation, and evolution, are the pillars of the significance of meiosis section. This cheat sheet table compares everything we just covered. Keep track of the differences. Meiosis I is reductional and separates homologous chromosomes, while meiosis II is equational and separates sister chromatids. And always remember, crossing over is exclusive to Pachytene. Meiosis is the physical mechanism of heredity. By reducing the genetic code exactly in half while simultaneously shuffling it, our cells ensure the continuity of our species while driving the diversity that helps life survive.